Welcome to everyone for this Idea Accelerator showcase. It is a real joy always to see the projects that students have worked on. The work we do with BioBuilder is really intended to do exactly what you have accomplished. That is to give students a chance to see biology as something that they can build with and do things with. And we have lots of great examples from that for today. This online program has arisen as a consequence of the COVID efforts that, the COVID conditions that we are living in, and we've moved all of our programs to online. But we are very excited about this program and the projects that have come up from it. And I am sure that we will continue to run this Idea Accelerator, this 3-week program in biodesign, as we move out of the COVID era and back into more normal and in-person effort.
Patients will take a pill containing a chemical inducer when they feel the onset of migraine. We want to engineer neurocells that will have the ability to detect high levels of the CGRP protein in the brain. We're focusing on CGRP proteins because they bind to specific receptors in the brain that cause the symptoms of migraine. The organism would then produce a ligand, which is just a molecule that will bind to those receptors and block the CGRP proteins from binding. And that would ultimately stop the symptoms of migraine. And as for our parts, we will have a constitutive promoter. So this promoter will be unregulated in order to make the effects long-lasting. We'll also have an inducible promoter to turn on only when there's too much CGRP, and a ribosomal binding site in order for the proteins to start translation. So to break down how our bacteria would work, the main input would be a low insulin level or high blood sugar, and then the output would be the bacteria would produce insulin, and it would also blow up to let us know that it's working. So further breakdown, our first input, well, technically we have 2 inputs, but our main input is a low insulin. Then our first output would be the insulin production in the pancreas cell, and then like the body itself, and then the organs will glow on top of that.
The topic we chose was a bacteria that detects the amount of gas coming from a certain area in the pipes. And what the bacteria would do would change the color or stain this particular part of the pipes that were leaking gas.
And basically our 2 devices are the first device being the gas detector and the second being a feature for generating a glow.
The first device will have a constitutive promoter, as it will always be on, detected for gas. If gas is detected, the first device will generate a protein for the second device to trigger and generate a glow. Otherwise, the second device will stay off.